Okay, so basically hereditary and variation. So we're going to be focusing on this particular topic, and basically when it comes to hereditary and variation, it's basically talking about how genes are passed on to another generation, to the next generation, and so on and so forth, right? And obviously that's hereditary, right? So how genes are passed on, and then variation really just talks about genetic diversity and basically how species can be different as they move from generation to generation, what changes they make, and how does this variation basically benefit them. So basically, hereditary and variation is a part of both the syllabuses in the CSEC biology syllabus, and it's also a part of the CSEC HSB syllabus, right? So that's the plan. So it's kind of going to be like a quick short course, not short course, like a crash course, and basically hereditary and variation. So just going over the main pin points that we need to mention if we're going to be talking about hereditary and variation. So what's the plan? So the plan is hereditary, we're talking about DNA, we're talking about genes, we're talking about alleles, chromosomes, and different types of genes. We're going to be talking about cell division and obviously variation, and then monohybrid, dihybrid crosses, and then complete, incomplete, and co-dominant. So that is the plan with all of this stuff. So, as we said before, hereditary. So basically, we're talking about how genes are passed on from parents to offspring, right? So, for example, let's just say my parents, or let's just say, yeah, my parents have brown hair, right? One parent have brown hair, and then one parent have black hair, right? And then I am the offspring, right? So hereditary is all about how those genes are passed on. So one of my parents have brown hair and the other one have black hair. So what color hair do you think I'm going to have when we mix? Because this is what we're talking about. This is a little bit of genetics, right? So we're talking about genetics when we talk about hereditary and variation, right? So we have to talk about that. So basically, what hereditary and variation and genetics, all of those, they're trying to explain how the offspring end up the way it end up basically. So the question is, if one parent have black hair and one parent have brown hair, which color hair the offspring is going to have? So basically, that's what hereditary explains. So basically, it's the passing on of genes from parents to offspring. And then variation talks about the differences in those traits between individuals or groups. So as I said before, variation is really and truly talking about the differences, right? So for example, because okay, one way to remember it, when you say the word variation, think different, think genetic diversity, because that's basically what variation is talking about, right? So for example again, and we can, as we go along, we're going to explain these things a little bit better, or we're going to learn the biological concepts that can explain these things a lot better. So for example, if we're talking about variation, let's just say maybe diabetes or something running at the family, and let's just say the next generation come and they don't have diabetes. So you're probably say, that's different, right? And basically this difference in the genes or genetic makeup is what makes these offsprings, the next generation, a lot more stronger. It makes them less susceptible to diseases. It just overall helps them to live longer because they're different and they're able to adapt to the environment differently, right? So that's the main difference between hereditary and variation. But overall, these two concepts are what helps us to understand how traits are passed on from the parents to the offsprings. Right, so now if we're going to be talking about anything hereditary, if we're going to be talking about variation, if we're talking about anything genetics, we have to talk about DNA, because this molecule right here is the backbone. It is the backbone, it is the foot, it is the everything. It starts from here, right? It starts from here. Now, basically, every living organism has a DNA. So it's either they have DNA or they have RNA, but RNA is only in viruses, right? But we're not talking about that right now. What we're talking about is specifically DNA. So it's deoxyribonucleic acid, right? This molecule, deoxyribonucleic acid, carries the genetic information, right? It carries the genetic instructions for basically our entire being, right? It is what we make up of. So for example, if you ever hear like a person saying, oh, let's just say that a couple, right, or they were a couple, then break up, and then the male say he want a DNA test, right, for the child because maybe you don't believe the mother or something. But let's just say the father want a DNA test. And that is why DNA is so important, because why you think him want a DNA test? Why him test the DNA? Because that is where the genetic instructions are, right? And if a person, and within families, you share DNA, right? You share some form of DNA with your parents, with your cousins, with, for example, your cousins, people like that. It gets lesser and lesser, right? But for somebody as close as like a father or a mother, you will find a good amount of the same genetic makeup, right? The DNA. So basically, it carries the genetic instructions for development, functioning, growth, and basically reproduction. Because let's just say that one of my parents them are really short, right? And then me come out and be really, really short. Why me come out really, really short? Because one of my parents were short, and it's because that gene got expressed in me, right? So we're sharing the same DNA, and that is why I look the way I look. That is why you probably look like your mother, you look like your father, you look like somebody, but you look like somebody in your family. It's all about the DNA, the deoxyribonucleic acid, right? That genetic molecule that codes for everything down the line, right? So without DNA, you are not you, right? It starts from here. Now, if you look at this diagram, so the structure of DNA is known to be a double helix, right? So it's a double helix, it's two strands, right? It's a two-stranded molecule. That's how we say a double helix. So that's how the structure of DNA would basically look. Now, if we're talking about genes, now genes are also very, 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 very important, because genes are basically a segment of DNA. So if you could look at this diagram right here. So before we say that this is a double helix, this is the DNA molecule that is within us, right? It is what because this is me. We need DNA for anything, for like cell division, right? The fact that we grow, we need it all starts from DNA. And maybe if you remember the process of cell division, if you remember mitosis or you remember meiosis, you would, and we're going to talk about that later on, but you would remember and know that within the nucleus, that is where you find the chromosome, right? Right, and inside the chromosome you obviously have the DNA, and then DNA you're gonna find a gene. So the gene is basically a segment of the DNA molecule. So if this, for example purposes only, if this was the entire DNA molecule that we had inside of us, that small section, that small segment would be the gene, right? And it's basically a fundamental unit of hereditary. That's what the gene is, right? So it contains instructions that are specific to make the specific protein or the specific RNA molecule, but RNA is mainly come up in viruses, but obviously it plays a role in what do you call it now? We can't remember right now, but RNA is also important in our DNA replication process, yes. So that's some molecular biology business, but we're not getting to that. But yeah, so genes help to dictate an organism's traits at the end of the day. So if you look at this is what I was saying. We have the nucleus. This is the cell nucleus. You have all these chromosomes inside of it. And on these chromosomes, you're going to find a DNA molecule. You're going to find DNA. And where you find a DNA, you're going to find the genes, right? So all of this come together in order to make us us, right? And remember, in our bodies we are made up of millions, millions, and millions of cells, because cells are really, 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 really small. This diagram is showing an animal cell. Look, can you imagine that? We're talking about DNA, and we're talking about genes, right? And look at this diagram. A cell can only be seen under a microscope, and then you're telling me that I'm telling you that after you see this cell under a microscope, it gets even more complex, because you have chromosomes in that nucleus, right? You have chromosomes in it, and you also have your DNA in it, and you have the gene, which is a segment of the DNA in it. So it's a really, really complex process that sometimes you probably can't wrap your head around the fact that, okay, the cell is already so small and so much is going on inside of it, right? But just remember that the gene is a fundamental unit of hereditary. So, and it's a segment of DNA. So once you see the DNA molecule, which is a double helix, just remember that a segment of that is a gene, and it codes for a specific trait, maybe blue eye color, maybe black hair, but it codes for something. Right, so the same chromosome that we were talking about, that is inside of the nucleus, right? And these chromosomes are basically the stars during cell division. So during mitosis or meiosis, they line up at the equator, they become condensed, they start out as chromatids and all that stuff, but they're basically show behind cell division. But just to get an idea of what your structure is like, so it's like a thread like structure, right? So basically this representation is when they are condensed, right? So when they are decondensed or uncondensed, it makes them a lot harder to see. It makes it a lot harder for us to see, right? When they are not condensed. So when they're condensed, they resemble this, but in all reality, they are made up of thread-like structures, right? And if we get a little bit more complex, like some nucleosomes and stuff like that, because it's literally a thread-like structure, and we can reel it all out. We can literally reel it. So this chromosome, if we take piece, I would just start is literally strands of nucleosomes packed together that make up the chromosome, right? But that's not the point. We just want to know at this level that it's a thread-like structure within the nucleus, and it carries genetic information in the form of DNA, because we know that. And by the way, DNA are also packaged with proteins and all that stuff in here, right? But we're not getting into any of that right now. But basically, the chromosome, and we know that the DNA can be found on the chromosome, and then we know that the gene can also be found on the chromosome, right? We know that. So that is why it carries the genetic information, because it carries all the structures that code for our genes. We have our gene, we have our DNA, everything is locked within these chromosomes, right? So now bear in mind, or let me just repeat again, that a gene is a segment of DNA. We said that before. Now we have different types of genes. So basically we can have a dominant gene, and we can also have a recessive gene. So a dominant gene would be a gene that is expressed, meaning that it shows up, right? And then the recessive gene would be one that is present but not expressed, right? So we say that dominant gene is represented by a capital letter. In this case, you're going to use capital B. And then recessive gene is represented by a common letter, and in this case we're going to use a common B. Now, how I would explain this is that, as I said before, we said that genes code for a particular trait. So now these B, and this is example purposes only, because if we want to represent the gene, we can use anything. We can use a big A, a big B, a big C. It depends on what trait we're coding for, right? So in this situation, this big B is going to represent black hair. Me, I'm going to make it represent black hair, right? But if you get a question or something like that, you might see C, you might see big R for red flowers. It depends on little r for white flowers. It depends on the question, it depends on the gene and all that stuff. But in this example, just know that the dominant gene is always the capital letter and the recessive gene is always the smaller, the common letter. But in this case, we're going to let capital B code for black hair, right? So this is going to be black hair, and then the recessive gene right here, the recessive gene, we're going to let the recessive gene code for brown hair. So it means that in this situation, and bear in mind that you get two, right? You get one from each parent. So it means the example I'm used before, say one of my parents have black hair and one have brown hair. But in this situation, brown hair is the recessive gene, right? So let's just say that my father did have black hair and me get this gene from him, capital B, and let's just say that my mother have brown hair and me get this recessive B from her, and because it's recessive, so basically these two B's, the capital B and the common B, both of them make up who I am right now. That is the gene that me end up with, right? Because we get one from each parent, right? And if we go back to sexual reproduction and we go back to fertilization, we know that it's both parents involved, so the offspring is going to be determined by the genes that them get from both parents, right? If it was asexual reproduction, there's just one parent, so whatever the parent have, that you get back. There's no question or none in it. But since it is sexual reproduction and we know that we're going to get one gene from each parent, and I said before, if my father have black hair and my mother have brown hair, but the gene that she passed on to me was recessive, right? And the black hair gene, the black hair trait which my father gave me is dominant. So when they made me, and this is me now, and me end up with a capital B and a common B, what do you think would be my hair color now, right? So this codes for black hair, this codes for brown hair, and me get both of them. What color hair do you think I'm going to have if my parents give me both these two genes? My hair color is going to be black, right? And it's going to be black because it's the dominant gene. It means it overpowered the little b. The little b is not going to be expressed. It calls for brown hair, so it means that, let's just say in the future, if me meet somebody, if me have some kids, one of my kids then probably will end up with brown hair, right? But at this stage and how they made me have black hair because that is the dominant gene. The black hair, dominant hair, the brown hair is very much recessive, right? But one thing to note is that a recessive gene, when you have two recessive genes, it will be expressed. So for example, if me never have a capital B from my father in this example, and we have if there were two common B's here, that may get one common B from one parent, one common B from the other parent, so brown hair come together, obviously we're going up with brown hair, right? Because the only time a recessive gene is expressed is when both of them come together beside each other. That's the only time, right? That is the only time it's going to be expressed. And the next thing to note is that with the recessive gene, it can show up later on. So you might pass this generation and next generation, and you just say you just don't see anybody with the brown hair, right? But it does not mean that the person is not a carrier, and it does not mean that the person does not have the gene. It just means that their gene is recessive. That means it's like dormant. It did it, but it cannot be expressed. And so if you keep on if an entire family keep on reproducing, meet people and reproduce new offspring, and let's just say that a pair dominant, right? Pair dominant gene the child gets, then the brown hair does not go expressed, right? But it does not mean that the gene is dead. If one person in the family buck up on somebody who have the recessive brown hair, and then brown hair and brown hair come together, the child again up with a brown hair. So that is why in some situations certain things can skip much generations and generations and generations, and then 10 generations later, something that you think they did out long time a show up back, your great grandkids or something like that, right? So that's basically how the dominant and the recessive gene works. Okay, now then you have so if we're talking about genes, right, they can also be homozygous and they can also be heterozygous. So we said before that the recessive one is not the recessive gene is not expressed unless there are two of them together, right? So if a big B is before the little b, that little b is not going to be expressed, right? So homozygous, which means same, means that two alleles or two genes, basically it's just basically it just means that both of them are the same. So in the example here we have 2b, so you have big B, that's homozygous because both alleles are the same, both genes are the same, right? Big B. And then you have heterozygous, which means that different. So it means that one of them are different. So you have big B, little b. And obviously recessive, right? You have little b, but in this situation, if we put another little b right here, if we put another little b, if we put another little b right here, that would be homozygous recessive, right? So we can just say homozygous recessive. Right, so that could be homozygous recessive. So you have homozygous dominant, you have heterozygous, and then you have homozygous recessive. Those would be the three, right? Those would be the three main ones. Now, if we're comparing the genotype now versus the phenotype, so basically when we talk about genotype, we're talking about, and an easy way to remember this is the word geno. Just remember genetic because both of them start with G. So the genotype is really talking about an organism's genetic makeup, and the phenotype is talking about the physical characteristics that me and you and everybody can see. So genotype, genetics, and then phenotype, physical characteristics, right? So in this situation, if we had a genotype, we have big B, and we say that B, the big B equals our black hair. So this is the genotype, right? This is the genetic aspect of, yeah, big B, and then the phenotype would be the black hair color. Now, so basically, alleles, they're basically a form of a gene, right? So there are a pair of genes that occupy a specific location on a particular chromosome and they control the same trait. So alleles, they are a form of a gene, and examples would be hair color, eye color, flower color. So they're just like different versions of a gene, right? So that is what alleles are. Then we want to find out why is variation important, right? So we said before that all of this, the entire point of all of this is to really see how genes are passed on from one generation to the next. But how does something like variation drop into this, right? How does variation drop into this? So basically we talked about that variation, we're really talking about what is different between the generations or what is different between the species, right? And so basically variation is extremely important. Why? Because it leads to genetic diversity, right? It's necessary for survival of any living organism, and basically it helps organisms to adapt to new environments. It builds up like resilience so that a species does not become extinct, and it also makes organisms less susceptible to diseases, right? So imagine, and by the way, the variation would come from sexual reproduction and only sexual reproduction. Something like asexual reproduction, if we have a flower, it produces asexually, right? And it is create a bunch of offspring just like that. Let's just say that the flower is susceptible to a particular disease, right? And then it will have like 20 offsprings that exactly like it. What's going to happen is that if that disease that can basically kill the parent, I will kill off the entire 20, right? And that puts the species at risk for extinction because them can't adapt to the environment. Them have the same exact trait, them have the same exact genes, them can't adapt to the environment. So it means that the parent are dead and all of the offsprings are going to die as well. What I mean extinction, right? So that is why variation is important, because even for human beings, if you look at human beings in the sense that what if all of us were exactly the same? For example, like a flu come, you know that some people can actually die from flu, right? Because if the immune system really weak, they can die. So like for even people with HIV and stuff like that, and the virus weakens their immune system. So for example, if me catch a little flu, we can get over it. And let's just say somebody with HIV, them catch a little flu, and because we know that their immune system is very weak because the virus take over. Basically, if they get a little flu, it's not a little flu for them because it will affect them body way more than it would affect me with quote unquote normal, right? So what's going to happen is that yes, so let's just say it affect them body really bad. So it makes them more likely to die because you might say, oh, let me just die from a little flu. But listen, if their immune system is really if the immune system really down and them catch a flu, I eat that for them because their body cannot fight back. The T cells them cannot fight back. They cannot respond, right? Because the virus hijack themselves. So what if that person with HIV, what if everybody was like that person and basically everybody have the HIV, it means that just like how that person could die from it, it means all of you could die from it too. But in this situation, we're talking about variation and we're talking about diversity. So it means that different people are different, right? Different organisms are different, different species are different, right? And it's necessary for survival because the truth is that if a disease come right now, some of you go dead. Some of we go dead, some of we go live, and some of we depend on the borderline, right? But it's very important to have variation. It's necessary for survival. And the HIV example, we only use that example because it comes to my head off the bat, but this is not anything related, this is not anything stigma related. We just want to make that clear. But at the same time, HIV does weaken the person's immune system and right? So it's not necessarily the HIV that kill the person that can kill somebody, but it's just the what it does to the body, meaning that it hijacks the body cell. It's a virus, and it weakens the immune system significantly, where T cells cannot fight back. So that is why variation is important. Or somebody with asthma, basically people have different illnesses, right? Variation, and for example, maybe as I said before, maybe diabetes run in the family, but meaning you're more likely to get it. Not that you can have kids and it pass on just like that, but you're more likely to get it if it already run in the family. So it just means that you have to be more careful with your diet and stuff like that. But even in that situation, the fact that just because your grandmother did have it, don't mean your mother got have diabetes. Just because maybe if your mother have diabetes, it don't mean that you must necessarily get it, right? So that's really what variation is and what it's basically talking about, right? And we said before that if we're talking about variation, it will come from sex reproduction, the fusion of male and female gametes, some fertilization have to take place, right? And another way that variation can come about is through a mutation, right? So mutation meaning that there's a change in the DNA sequence, right? So for example, during maybe cell division, we know that during cell division, during the S phase, that's where DNA replication takes place. Let's just say something went wrong, the spindle never attached properly, something go wrong, a mutation happened, right? And then bam, you realize say you're gonna be different, or I don't know, something gonna change though because it never um line up the way it should, or it never attached the way it should. So that's basically like a what's the word? It's more like you're more likely to have an abnormality, yeah, something like that, abnormality. And another way that you can get like a mutation happening, right, is through mutagens such as radiation. That is why them tell us to stop microwave food, cook food, stop microwave food, stop be close to like radiation, heat, anything like that, because it's not good for us. So that's why they say that. And not all variation is good, as you can see based on the whole radiation thing, because you know that can kill you, and radiation can trigger the cancer cells and bam, you have cancer, different things. So it's not all variation is good, but it can basically change things, just know that. Okay, so now cell division, mitosis, and meiosis. So we're going to focus a little bit on cell division, and Not going completely in detail about it, but we're just gonna run through cell division quick and fast. But basically, there's a more in-depth, I think we call it in-depth video about cell division, so you can definitely go and look at that, both meiosis and mitosis, so you can look at that. But now we're just gonna run through really quickly, right? So as it relates to the stages, well, okay, so we're not going to go through the stages of interphase. Yeah, that's what I mean to say. But we're going to focus on just PMAT. So basically, cell division is the process by which new cells are formed, and the main two types of cell division are mitosis and meiosis, right? Now, there's a difference between mitosis and meiosis, so it's not just one cell division take place in our body for everywhere, right? No, there are two main types. We have mitosis and we have meiosis. Mitosis, that's where our body cells slash somatic cells, they divide. So the word somatic is really talking about body cells, so same thing, body, somatic, same thing. So in mitosis, division of body cells or somatic cells to produce two diploid daughter cells. So we know that the end result of mitosis is going to be 2, and it's represented by 2n. Meiosis, on the other hand, is a specialized type of cell division, right? Because it only focuses on our sex cells, right? So this is how we basically reproduce to have like actual offsprings. No, mitosis is really for like bodily repairs, maybe you get a cut, cell division, feed heal and stuff like that, right? So repair and yeah, just mainly repair and build up cells, maybe replace worn out cells. So it's really repair and replace of worn out cells in the body. That's what mitosis is. While meiosis specifically, we're talking about offsprings in this situation. And the end goal of meiosis is going to be 4 haploid daughter cells, and the haploid is represented by a N. So this is the process of mitosis. So basically, how I remembered it is PMAT. So prophase, metaphase, anaphase, telophase, and then you have cytokinesis. Now, I said PMAT. So why we include cytokinesis? We're going to explain that. But even before we talk about these stages, we have to talk about what happens. We're not going into too much details, but we're just going to brush over it. Interphase. So interphase is that preparatory stage that the cell is basically in before it starts to divide. Now, the cell spends a lot of time, in fact, spend way longer in interphase than it actually spends in the actual mitosis process, right? And interphase comes before both mitosis and meiosis because that's where it's like a resting stage. The cell is just in there growing. It's just getting ready to divide. No, I'm never gonna mention the stages, but we just brush over them. So basically in interphase, we're going to have DNA replication taking place. We're gonna have the cell growing and then growing some more. That's really interphase, right? So the cell spends a lot of time in that stage. Now let's start focusing on mitosis. So after interphase done and gone and the cell enters mitosis, what's going to happen is we're going to start with prophase. So what's happening in prophase is that these chromosomes are going to become condensed. So as you can see right here, these two are the chromosomes in this situation, right? And this example only shows 2, right? But there can be a lot in there. But this example only shows 2. But this is where the cell is going to, and the chromosomes become condensed, and then you'd also see the central, what's it called, the centrioles. So the centriole or the central, let's just say centrosome, because the centrosome has in the centrioles, the centrosome, which is basically going to be like like a circle or something. They're going to be on opposite ends of the cell. This diagram don't really show it, but it's going to be centrosome. It's going to be an opposite end of the cell. Centrosome is going to be there. Is going to separate the opposite ends of the cell. That's going to happen, and the chromosomes become condensed. So that's really what's happening in prophase for the most part, right? And the reason why they're becoming condensed is because before they were not condensed, they were uncondensed, they were decondensed, they were not showing, right? And now they're showing, so they're condensed. So that's prophase. So as we move into metaphase right now, by the way, in the middle of these chromosomes, so basically one of these are called a chromatid. So these are two chromatids joined together by that circle ball. The circle ball is called a centromere. So the centromere is holding the two sister chromatids. So individually they are chromatid. One of them is a chromatid. Two of them is a chromosome. So when they're joined together by that centromere, the little ball in the middle, it becomes a chromosome, right? So back to metaphase. In metaphase, we said before that we had centrosomes at the opposite ends of the cell. So this diagram I'm not sure, but it would be like a little circle ball, right? Circle ball, circle ball. And what's happening in this metaphase is that the chromosomes become lined up at the equator. So this right here is middle equator. It lines up right here, and from these centrosomes or centrioles, we're going to have these spindle fibers extending. When these spindle fibers extend, they are going to basically attach themselves to something called a kinetic core. So the kinetic core is at the back of the centromere, so it's going to attach to the kinetic core, which is on the centromere right here. So these spindle fibers, they extend, they attach themselves, and we have the chromosomes lining up at the equator. Those are the main stuff that are happening in metaphase. As we move down to anaphase, what's happening here is that, and how you remember, by the way, just a way to remember, prophase, pro means before. So before, as you can see them just in there and I really don't much, and it's become condensed. And then metaphase is from a middle, they line up in the middle. Anaphase means away. So in this stage, these chromosomes are being detached, right? So when you have like a force, two opposite forces, it's going to split it apart. So in this case, what's happening is that the sister chromatids are split apart again, and they're pulled to opposite ends of the cell. That's what's happening here, right? Because two of them were chromosomes, and now they're pulled apart, going to opposite ends of the cell in anaphase, right? So they're pulled away from the equator. Now, what's happening in telophase is just that after they've gotten to opposite ends of the cell, we're going to have a like a membrane forming around them, right? Actually, yeah. So basically, they have like its own individual like nucleus forming around it. You know, when if that would be the right word, well, let me just work with it right now. But they can, yeah. So they go to opposite ends of the cell, and as you can see, yeah, I don't want to mention that, but yeah. So they're pulled towards the end of the cell, and you have equal amounts on both sides in telophase. And then cytokinesis is what is going to split them into two. So these two are going to become the daughter cells. Two of them are becoming daughter cells. So the goal behind the entire mitosis is to produce two daughter cells at the end of the day. And as you can see, we end up with equal amounts in each daughter cell. So that is the goal of it. And also in telophase, this is where the we call it the nuclear membrane. So the nuclear membrane starts building up. So that's what's happening here. The circle around it, the membrane is the nuclear membrane around it, right? So it's a nuclear membrane, and the chromosomes start becoming decondensed. As I said, this diagram is not the best diagram, but yeah, they become decondensed. So you see how visible they were? That means that they start not being as visible, right? So the end goal is kind of like for daughter cells to look similar to what we started with initially. But what's happening? Nuclear envelope or nuclear membrane rebuilds, chromosomes become uncondensed in telophase, and then at the end we end up with two separate daughter cells. That's basically mitosis. Then in meiosis, it's basically way more complex. It's complex, but at the same time, it's the same thing. Okay, so what's happening is here in the prophase. So what's happening is we have PMAT twice, right? We have PMAT twice. So it means that the cell is going to have a prophase 1 and a prophase 2, a metaphase 1, a metaphase 2, and a phase 1 and a phase 2 is going to have a telophase 1 and a telophase 2, right? Because at the end of the day, we're going to end up with 4. That's the goal, right? The goal is to end up with 4. So in mitosis, we ended up with 2. Meiosis, so we basically do mitosis twice. This is, we can just kind of look at it like that. And yeah, we do it twice. So we're gonna have double the daughter cells at the end of the day. So first in prophase, and basically the first round of meiosis is basically just mitosis, right? So you're going to have, but bear in mind that meiosis is the division of sex cells, right? So we're talking about genetic variation and all of that, and we get one from each parent, right? So that's the difference with mitosis and meiosis. So prophase 1, the nuclear envelope is going to break down, chromosomes are going to be condensed, and then something that did not happen in mitosis, which is very important, is crossing over. So crossing over is basically where these diagram don't really show it and so good, but basically if you look at this one where you see the red have the blue on it, this one with the red have a blue on it right there, that would be crossing over where piece of like a segment off of one chromosome cross over and go on the other one, right? So that's how we get the genetic variation. So the nuclear envelope is gonna break down, chromosomes become condensed, crossing over takes place in prophase 1. Metaphase 1, same thing, line up at the equator, line up in the middle, and then we have the centrosome, what I talked about earlier, and the centrioles, right? So in metaphase, the centrosome is going to they're going to extend the spindle fibers, chromosomes are going to line up at the equator, right? Now there's something that is very important here. So in metaphase 1, you can see them line up at the equator, and then if we just jump ahead a little bit and look at metaphase 2, they're also lined up at the equator, but what's the difference is that they're lined up in different directions. As you can see, you see it? They're more horizontal over here, so them very vertical. They line up differently. And also, the next thing we need to make mention of is that we don't just have two chromatids joined together by a centromere forming one chromosome. What we have going on is we have homologous chromosomes in the nucleus. That's what we have going on here. We have homologous chromosomes, and these homologous chromosomes can only be found in meiosis. You cannot find them in mitosis. And as we said before, because we're getting from both parents, right? So that is why we get chromosomes from both parents, right? So at the end of the day, we have 43 chromosomes in total. Also, right? No, we have 46. Sorry. We have 46 chromosomes in total, 46, 46, and we have 23 chromosomes from each parent, right? 23 from each parent, 23 and 23 make 46. So we have these homologous chromosomes, they become condensed, nuclear envelope breakdown, crossing over take place. That's how we get the one version. That's how we get one way of getting genetic variation. We soon tell you what the other one be. And it's within these steps. So the metaphase 2, they line up at the equator, spindle fibers extend, bump to the joint to the kinetic cores at the back of kinetic cores on the centromere, right? So spindle fibers extend, attach themselves, anaphase the chromosome. So before homologous, or we could also call them a tetrad. So that's basically 4 chromatids, 2 chromosomes, 4 chromatids make up the homologous pair, right? So here when we have pulling apart is we have one chromosome and so, one chromosome and so, right? And you can see the genetic variation. You can see what happened because of crossing over, that we have different color upon different color, right? So that's what happened. Then in anaphase, they're being pulled opposite ends of the cell, And then in telophase and cytokinesis, the nuclear envelope is going to reform, chromosomes become decondensed, meaning they're going to be less visible now, and obviously the spindle fiber is gone, and it's going to split into, it's going to create a little hunch in it, like kind of like split into two, right? So now prophase 2, right? But bear in mind, so even though it's splitting into two, it don't split what is it? It don't really necessarily split completely, right? So it now got the pinch off right here, so not here, so not time it. But in prophase 2, it's basically the same thing that gonna happen. Chromosomes will become condensed, and you know the centromeres are going to separate the opposite ends of the cell. Metaphase 2, these are chromosomes lining up at the equator. In metaphase 1, we had homologous pairs, we had tetrads lining up at the equator, right? We had 4 chromatids, 2 chromosomes. Here we have 2 chromatids and 1 chromosome, right? Lining up at the equator, and as you can see, in metaphase 2, they line up more vertically. Anaphase 2, the chromosomes are both opposite ends of the cell, right? And then we're going to have telophase and cytokinesis, where basically nuclear envelope is going to break down, become decondensed, meaning less visible, and then you're going to split. So at the end of the day, when the cell finally does cytokinesis and splits, we're going to end up with 4 daughter cells, right? So cleavage barrow is right here, but it now completely split. So what's going to happen is this would enter in over here. So I would run it again, I will end up with 4. The end of this diagram don't necessarily show the 4, but you get the point. Then the last thing we're gonna mention is really about well, this is not the last thing for what I think, but yeah, different inheritance. So basically how inheritance works. So as I said, the entire point of all of this is to find out how genes are passed on. So what's happening here, we're going to talk about the monohybrid cross first, and then we'll talk about dihybrid afterwards. So in a monohybrid cross, it's a genetic cross, right? So this is how we can kind of like predict the next generation, right? So it's about genetic cross between 2 homozygous individuals, right? Remember what we said homozygous meant, they must have the same thing. Now in this diagram, this will be the F1 generation, meaning the first generation. So if the parent had a child, this would be the first generation afterwards, right? The offspring is the first generation. We said homozygous. You can have homozygous dominant and also have homozygous recessive. So if you cross a homozygous dominant with a homozygous recessive, so if the one parent had black hair, so BB, which goes for black hair, which, well, not black, oh no. So if BB, forget, so we do a different example. Let's say we do flowers, my bad. So we're doing flowers in this example. So BB, so the big B is blue, which is dominant, and then the little b is white, which is recessive, right? So if we coded big B, which goes for blue, which is dominant, and then we code homozygous recessive, which goes for white, what's going to happen is that we're going to get 100% 100% blue flowers, right? 100% blue flowers, right? So as you can see before, this is the genotype. So it means that all of these flowers have the like the trait for white flowers, but it's not expressed because the dominant one overpowers it. So it's 100% blue flowers that I get, and then I get 100% for the genotype, you will get 100% big B, little b, and then for the phenotype, 100% blue flowers. So where things kind of get more differentiated with our monohybrid crosses when we go down to the F2 generation, and that's basically the second generation. Now we're using a different example. So what's happening here is we have 2 homozygous, well, not homo, 2 heterozygous individuals, yeah. So let's just say right here we have all of these individuals are heterozygous, by the way, right? Because they have 2 different genes, two different alleles, right? So yeah, so usually at the end of the F1 generation, you're gonna get heterozygous. Now when we go to the second generation, right, meaning heterozygous. So if an heterozygous crosses with another heterozygous, which in this case we're going to be doing big R for red flowers, and then big R little r for also red flowers, and then little r for white flowers. So red flowers is the dominant trait, and both these genotypes code for it, right? And then little r, that's white flower, which is recessive. So if we cross two heterozygous, meaning big R, little r. So that's the genotype, big R, little r, and big R, little r. So that means that we're crossing two red flowers, right? So it's two parents and both of them look red. What do you think could happen? But they have different genes, they have different alleles, and we say that alleles are basically different are basically alternative forms of a gene, right? So it can code for different things. So in this situation now, we have two heterozygous, both of these flowers look red, but if we want to predict the next generation, we can do a cross, and that's what we're going to do now. So the first child them have, and this is just a Punnett square that we basically use to do it. It's called a Punnett square. It's very easy. It don't hard. Just draw four box, and then you get the alleles of big R, little r, big R, little r. Because in this situation, it is heterozygous crossing with an heterozygous. We know that both heterozygous they code for red flowers. So now two flowers that look very red, we're going to cross them, right? And then the first child that I'm have, so this first 25% is going to be red flowers. So it knows, okay, the first child will come out completely red flowers, right? And them now gonna have no trait. Them not gonna have the trait for white flowers. Cause them not like a R. So it means say them fully red. Them not no trait for white flowers. Then the other two, which is 50%, we realize they both have the white flower trait that carry with them. So that's 50% for the genotype, big R, little r, and the phenotype is going to be red flowers, right? And then the last one is going to be little r, which is recessive, and it's going to go for white flowers. So at the end of the day, we have 1, 2, 3. We have 3 red flowers, right? We have 3 red flowers, and we have one white flower, right? So 3 to 1, right? And then for the genotype is going to be 1, right? One big R, 2 big R, little r, and then 1 little r, right? Right. So this should actually be 3. So it's not a mistake. It should actually be 3 to 1. So the phenotypic ratio is, we say that the phenotypic ratio is the physical characteristics. In this case, it's going to be the color of the flowers. So we're going to put 3 red flowers to 1 white flower. So if these flowers have 4 bikini, 3 of them are go red and 1 of them are go white. The rest of them are go, you get the point. And then the genotypic ratio is going to be 1 big R, 2 big R, little r, and then 1 little r. So that's basically the F2 generation, right? So as you can see, the first generation is usually very simple, really complex, everything 100%. So you're going to end up with 100% heterozygous. You're also going to end up with 100% of the dominant trait, which in this case was blue flowers. Now the dihybrid cross, which is a little bit more complex, right? So in a dihybrid cross, where you're going to be crossing two individuals, right? Which code for two different traits. So instead of drawing up that 2 by 2 squared little Punnett square, you're going to end up with a 4 by 4, right? So a 4 by 4. So we're going to end up with 16 boxes, right? 16 boxes in order to do a dihybrid cross. And the phenotypic ratio is going to end up with a 9 to 3 to 3 to 1. Genotypic ratio, you're going to end up with a 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1, right? So this is what is expected at the end of the day for the dihybrid. So then you have complete dominance, codominance, incomplete dominance, right? So you have that at the end of the day. So let's find out the difference between the three. So basically with complete dominance, It just means that only the dominant allele is going to be expressed. So for example, if you had two flowers, right? And we use a flower example, big R coded for red flowers, little r coded for white flowers. So the complete dominance means the offspring is going to come out completely red because that is the dominant trait. Incomplete dominance, neither allele dominates the other. So it means that if we had a white flower and we had a pink flower, no, let's just say we had a red flower and a white flower. If we had a red flower and a white flower and the fertilization, they mated, let's just say that, and the flower that we're going to get is going to be pink flower. So it means that both alleles are going to be expressed, and usually when that happens, it's going to give rise to a completely new trait or a different color, because obviously neither parents were pink. One parent did red, one did white. So how did we get pink? Because it was incomplete. It was like a blend of both colors. For codominance now, both alleles get expressed. So for example, if a red parent or a white parent, they have offsprings, and that one plant, we realize that it have both yellow, it have both white, and it have both red flowers, right? That's codominance because both of them are expressed in that case. Anyway, so that's basically it. There is a biology series which basically has the nervous system, more detailed cell division, reproduction. I can't remember the rest right now. Cells, cell structure and function, cell transport, different things like that. And then we also have the CSE playlist, right?